Thanks for the intro. Uh, so yeah, last talk of the day, so I'll try and keep it as light and entertaining as possible, talking about deep learning for fitness prediction. I can't see the slides. Oh. Wait, is this going back? Oh, okay, got it. Sorry, got a preview. So I'm going to talk about this method called ProteinNPT that I work with Pascal from the lab, and I'm co-supervised by Yarin Gal in Oxford and Debbie Marks in Harvard. So we were trying to develop a new method to improve protein property prediction and design with non-parametric transformers. So a central question in biology is how to go from genotype to phenotype and how one affects the other. And we can schematize this with this graph. So when we go from genotype to phenotype, we're trying to do prediction. So trying to predict a given phenotype from a genotype. And when we do design, we're trying to do the reverse task. So we know the phenotype, and we're trying to design a genotype that will have this effect. So in our case, we're looking at protein sequences, and the phenotype will be the function of the protein. And we're trying to find a model that can do this, so a generative model that can do both of these tasks together. And there's been a rich history in the Marx lab of trying to solve this problem with unsupervised methods, with a key insight that sequences capture evolutionary constraints. And so starting with multiple sequence alignment, you can just count up the amino acids at a given position. So these are site-wise factor models like PSSM, to more expressive models. And I want to highlight specifically Eve from the lab that is a very good variation autoencoder method for fitness prediction. And now we have the emergence of protein language models that are trained not on just one family of interest, but on across the entire proteome. So when we do unsupervised learning, we are not incorporating any of the experimental data that a lot of people probably in this room have carefully gotten from experiments. And so how do we integrate these required labels in our modeling? So we want to do this to predict and design for a specific function of interest that might be not what the protein naturally does, so that wouldn't be captured by these unsupervised methods. And we might want to select for something that isn't selected by evolution. So there's been classically two existing approaches in the literature to do this. The first one is from a paper here from Biswas, but it's been done by other papers. And what you do is essentially a two-stage process. You first train an unsupervised model, and then you get an evolutionary, you take the embeddings from the last representation of this model, and you train a top model on top of these representations with your acquired, on your experimental data. And then the other method, which was developed by Chloe Siu, was actually to just use a one-hot encoding representation of your protein sequences and add to that a score that is the likelihood that you would get from an unsupervised method. And then the same thing, you train a top model on top of these representations on experimental data. So the problem with both of these approaches, as I highlighted, you're doing the learning process in two steps. First, training an unsupervised method, and then trying to sort of steer the model towards predicting supervised method. But can we actually supervise the predictions? But can we actually do both at the same time? So can we learn from genotype and acquired phenotypes with one model that would be able to do prediction and design? So to do this, we turn towards this non-parametric paradigm. So essentially, What you're trying to do in a classical machine learning model is the method in the middle. So you have a parametric model that's trying to go from inputs to outputs, so from your x to your y, and you parameterizing this by a set of parameters that are going to be used for any x input. In the non-parametric transformer approach, essentially what we're trying to do is do the same task, but instead of just conditioning on your input, you can condition on your entire data set. And so you might learn more meaningful interactions between data points that you wouldn't capture with just this parameterized approach. And so by doing this, we can try and leverage evolution during the fine-tuning process so I'll highlight how this is done by the method. So here's what the architecture. Here's what the architecture looks like. So you take as input, if you start from the bottom, you take your protein sequence that you have embedded with protein language model. So that's the sequence embedder, the same as the whispers method. But you also take your auxiliary labels that you also encode, and you concatenate everything into one big table. And your task will be not only to reconstruct a given target, but you also might want to reconstruct masked amino acids. So you're combining essentially both of both of the objectives from the supervised method, so trying to reconstruct a given target, but also the objective from the unsupervised method, so trying to reconstruct a given amino acid into one framework. And you do this learning by applying successive row attention and column attentions. So one will reason about across data points, and the other will reason across attributes. So this combination of amino acid and labels. So yeah, as I highlighted, we mask a fraction of amino acids, and we also mask the target, and we have this loss that combines the two, and we have a parameter that weights the two, and this parameter is progressively annealed to increase the focus on the target prediction part of the task during training. And in a way, you can see this amino acid reconstruction part of the loss as a regularization term on the training objective. So at inference time, we form an input batch by concatenating a fraction of our test sequence, where we won't have the labels, and we're trying to get the labels for those, with training instances where we know the labels. And so this is and so these predictions are therefore dependent on both the learned relationships between tokens and targets, as well as homology across label instances. And it's this crucial part that we didn't get before with the previous methods. So this modeling approach has multiple advantages. First, we can very easily incorporate multiple target predictions. So we can just add a column in our data set with if we have more than one target, we'll have two columns essentially. And we can handle partially available labels. It's just we mask the label when we don't have it. And also compared to previous approach, we don't need this pooling operation where classically you would take the average token representation across your entire sequence. So to benchmark our method, we train one ProteinNPT model per DMS assay that we have in the protein gym benchmark. And we do multiple cross-validation split to try and compensate for, as was highlighted in the previous talk, of these relationships. So you have essentially, for a given position, you have a lot of correlation of, if one position is bad, a lot of mutations at that position will be correlated to each other. So to try and prevent that, we do different cross-validation schemes. One is random, and the two other ones are based on position. So each position will be assigned to one cross-validation split. They won't be shared across the splits. And those are the contiguous and modular. And essentially, we benchmark against the two approaches I talk about. OHE stands for one-hot encoding, and the embedding section is where, same as the Biswas paper, they train on top of embedding representations. And so we do a few ablations to highlight our design choice. I'll maybe just focus on essentially trying, the second one is looking at different sequence embedders, and we see that when we take better sequence embedders, we get better performance. So hopefully if we get an even better protein language model like POET, for example, a good recent model, we can retrain ProteinNPT on top of that, and hopefully we get even better performance. So we also do some ablations on looking at specifically this column-wise attention, so attention across data points that is specific to the non-parametric approach, and we see that it's also very useful and essential for performance gains. And we look at varying the number of sequences that we add at inference time, and essentially observe that, well, you get a boost up to 1,000 sequences. Then we also benchmark for the protein gene assays that have multiple labels, multiple mutants, sorry, and we perform quite well on those. And the performance lift we observe is also a brush to mutational depth. So as I said, we can use the method for multiple property prediction. So this is what we did here, trying to predict more than one target. And we can also use the method to get uncertainty methods. And we can do that by, instead of just doing the dropout method that we use in machine learning, we can vary the sequences we construct a batch with, and this will give even better uncertainty estimates than the traditional just dropout approach. And so to get to this part of the design process, so we try and imitate what we would want in the lab. So optimizing a given sequence for a property of interest, where we imagine for that within a DMS, we only have access to very few amounts of labels, and we have different rounds of acquisition where we're gonna select which mutants we want to acquire. And so we do 10 acquisition cycles, and we do this for each assay, and here I've plotted the average across all assays. And we see that we recover quicker high fitness points with ProteinNPT using this acquisition function that takes into account our fitness prediction and our uncertainty prediction. And to also highlight how you would use a ProteinNPT for design, we can condition, so both on the sequence and also on this on the fitness prediction. And so we take as input sequence the one with the highest fitness, and we try and see how well we could improve on that fitness prediction, on that fitness score, and we scored with an independent model, ESM1V. And we see that by doing this and resampling from our model positions, amino acid substitutions in our sequence, we get higher fitness distribution than naturally observed. And yeah, that's about it. Thank you so much for listening to me. And I want to thank everyone from the Marx Lab and the GAL Lab, and especially the Marx Lab for creating such a great atmosphere here in Harvard. Thank you so much.
Thanks, Ruben.
Hi, really nice talk. So I have a question about the training, right? So supposedly you didn't train from scratch, right? So the problem that there are not so many data points to train off, and we know that the quality of MAVE depends. So have you tried have you trained on all the protein gene data set, or you have done some filtering over it to remove something that has low, for example, correlation with predictor, that we know that it could be experiment, it could be predictor, but maybe they will reduce the quality of the prediction?
Yeah, so the results I showed were on training one model on each of the assays in Protein Gym. So there's 100 assays, more than 100 now, 250 assays. And so so essentially you train a new model on each one of these, and you'll have a varying amount of data points. For some assays, you have 50,000 data points, and for some you have a few hundred. And we did a few ablations on seeing how well the model performs with the varying number of data points, and it was actually quite independent in a way of number of data points. And there's probably more interesting stuff to look at, whether the performance lift is specific to certain assay in questions, or what exactly means that you would get a performance lift from supervised methods compared to unsupervised methods.
And following on this, so if I want to inference on a new protein, I should have like the experimental data to support it to train the model at this point.
Yeah, well, if you want to design a functional protein, I guess it depends what the goal is. If it's for design, definitely. If it's to characterize a new protein.
Have you ever tried like to do like a universal
Well,
one?
yeah, that's the, oh, you mean like, we only designed and then tested in silico with the ESM1V experiment, but we haven't
No,
done
it's just like, I mean, if you select like a good set, I mean, you could try to make something that, learn about fitness label, and starting from embedding, it creates actually whatever sequence you input and you set up.
And
have
then
you tried to do something like that?
Test it experimentally?
Without experimental, you train on the experimental data, and after you design on a new protein, like outside your training set. So you take more than one MAVE, you train more. Sorry, there is not.
Yeah, that's we can talk later, but that is essentially the experiment we do, the last slide actually.
Okay.
Yeah.
Thank you.
Do you have any recommendations for reconciling, basically adding multiple, labeled data sets to the training?
So training on more than one data set?
Training on multiple, like DMS
Multiple
assays or something like that.
Yeah, that's a good question. So on this approach, we didn't look at that. The problem with looking at different data sets is they need to have something in common. So it could be testing for the same function, I guess. But in this approach specifically, you need to be able to align your proteins. So it would only make sense for very, very similar, like proteins from the same family that have been tested.
Maybe this is
I can
totally
ask
stupid.
another question too.
So maybe this is totally stupid. I don't totally understand the details, but could you train it on, computational predictions? Would there be any value to doing that?
Yeah, that's a interesting idea. I'm actually testing some things on that now for antibodies.
But I guess you'd have to watch out for circularity potentially in that.
Yeah.
The other question I had is kind of an annoying one, which is how do you reconcile the fact that some of your mean squared errors weren't as high, although your Spearmans were for ProteinNPT, when you were doing the head-to-head comparison against some of the other models?
Yeah. Yeah, I think it maybe goes back to the point before about how the metrics you use are so important. So I don't know. Good question. I guess you could look into more details, exactly why you get better Spearman on some, but still the improvement in MSE is not that great. Yeah.
Are you looking into any other architectures at the moment, or is it mainly transformer, this one model you're looking at?
Yeah, transformers are pretty good for everything this is purely sequence based. So I think there's been some good history with transformers, like in the Marx lab, for example. So I think transformers for now. But now diffusion is all the craze, so maybe we'll have to change soon.
All right, well, if that's all the questions, Thanks very much, Ruben, for a very interesting talk.